Now we move on to specification number 10. This one is about the functions of digestive enzymes. We need to describe what each enzyme does, along with its substrate and the end product. This part focuses on digestive enzymes. It asks us to describe how different enzymes work, what their substrates are, and what products they form. In other words, we're looking at how food molecules are broken down step by step, starch into sugars, proteins into amino acids, and fats into fatty acids and glycerol. The key enzymes we need to cover are amylase, maltase, protease, and lipase. The first enzyme we'll study in digestion is amylase. Amylases are produced in two main places, in the mouth and in the pancreas, where they're then secreted into the duodenum. The job is to break down starch into smaller sugars. In the alimentary canal, amylase is released both in the mouth and later in the duodenum from the pancreas. Here, it converts starch into maltose, which is a disaccharide. That maltose doesn't stop there, it's broken down further into glucose by another enzyme called maltase. Maltase is found on the membranes of the epithelial cells that line the small intestine. As you can look at the diagram, you can see the process. Starch molecules are acted on by amylase, producing maltose molecules, which are then broken down into glucose. The next enzyme we'll look at is maltase. Maltase has a very specific job in carbohydrate digestion. It breaks down maltose, which is a disaccharide made up of two glucose molecules, into those individual glucose molecules. Once maltose is converted into glucose, the glucose can then be absorbed through the walls of the small intestine and passed into the bloodstream. From there, it becomes a vital energy source for the body, powering many different cellular processes. If you check the diagram, you'll see the process, maltose molecules, acted on by maltase, producing glucose molecules. Now let's move on to proteases. Proteases are a group of enzymes that break down proteins into amino acids, and this process happens in both the stomach and the small intestine. In the stomach, the main enzyme is pepsin. Pepsin works best in acidic conditions and starts breaking proteins down into smaller chains. Then, in the duodenum, protein digestion continues with trypsin, which is produced by the pancreas and works in alkaline conditions. Each of these enzymes has its own optimum pH, acidic for pepsin and alkaline for trypsin. Working together, they make sure proteins are fully digested into amino acids, which can then be absorbed in the ileum. In the diagram, you can see how a protein molecule is broken down step by step into amino acids with the help of protease enzymes. The last enzyme we'll cover is lipase. Lipases are produced by the pancreas and secreted into the duodenum. The job is to break down lipids, that's fats and oils, into fatty acids and glycerol. This breakdown is important because it makes fats small enough to be absorbed in the small intestine, allowing the body to use them as an energy source. Lipase works by breaking the bonds between fatty acids and glycerol. But before this happens, bile from the liver emulsifies the fat, breaking it into tiny droplets. This increases the surface area and makes lipase much more effective. If you look at the diagram, you'll see how a triglyceride, or fat molecule, is acted on by lipase to produce glycerol and fatty acids.